Damage. Furthermore, recent data indicate a favorable effect of flavonoids on neuroinflammatory events. Several reports also support the concept that flavonoid intake inhibits certain biochemical processes of brain aging, and might thus prevent to some extent the decline of cognitive functions with aging as well as the development or the course of neurodegenerative diseases. Schmidt-Schillig, et al., 2005. However, more research is needed to assess the true impact of flavonoids on brain aging. Saponins are steroid or triterpenoid glycosides, common in a large number of plants and plant products that are important in human and animal nutrition. Several biological effects have been ascribed to saponins. Extensive research has been carried out into the membrane permeabilization, immunostimulant, hypocholesterolemic, hypoglycemia, antioxidants and anticarcinogenic properties of saponins. George, et al., 2002. These structurally diverse compounds have also been observed to act as antifungal, antiviral and anti-aging agents. Table 1, Rasayana and Medrasayana drugs in Ayurvedic literature. S.No. Botanical nomenclature of plant, Ayurveda nomenclature, family, part used, pharmacological. Action for cure of AD. 1. Areca cateche, L. Supari, Ericaceae, fruit endosperm, M1/M3. Partial agonist. 2. Bacopa monnieri, L. Brahmi, Scrofulariaceae, whole plant, antioxidant, anti-ache, nerve tonic, cardiotonic and diuretic. 3. Celastris paniculatus, L. Jyothishmathi, Celastraceae, seeds, antioxidant, sedative and anticonvulsant, reduced monoamine. 4. Centella asiatica, L. Mandakparni, umbelliferac, roots and leaves, sedative, antioxidant, antidepressant, cholinomimetic, anti-ACHE immunomodulator. 5. Curcuma longar, L. Hydra, Gingeraceae, rhizome, antioxidant, anti-inflammatory agent. 6. Nardostachys jatamansi, L. jatamansi, valeriancoac, roots anti-ischemic, antioxidant, anticonvulsant, and neuroprotective. Activities, increase monoamines and inhibitory amino acids. 7. Withania somnifera, L. ashwagandha, solanaceae, roots. Immunoregulator, antioxidant, anti-stress adaptogen, neuronal regeneration, anti-ACHE. Ayurvedic nomenclature. Supari. Family. Ericaceae. 3, 5. 6. M/Ma partial agonist. Experimental study. In the present series of study, the three plants Bacopa monnieri, Curcuma longa and Hippophae rhamnoids have been investigated for their protective effect on aged brain. Brief description of the selected plants. Bacopa monnieri. Bacopa monnieri, a member of the Scrofulariaceae family, is a small, creeping herb and grows naturally in wet soil, shallow water, and marshes. The entire plant is used medicinally. 37, Bone K, 1996. Compounds responsible for the pharmacological effects of Bacopa include alkaloids, saponins, and sterols. Many active constituents, the alkaloids bromine and herpestine, saponins D-mannitol and hersaponin acid A, and monnurin, were isolated in India over 40 years ago. Other active constituents have since been identified, including botulic acid, stigmasterol, beta-cytosterol, as well as numerous bacicides and bacopasaponins. The constituents responsible for Bacopa's cognitive effects are bacicides A and B, Chakravarti et al., 2003, Ho et al., 2002, Mahato et al., 2000, Chakravarti et al., 2001. Figure 4, Structure of active molecule of Brahmi, bacicide A. The plant extract and its isolated bacicides have been extensively investigated in several laboratories for their neuropharmacological effects, and a number of reports are available confirming their nootropic action. Studies have established that the treatment with the plant extract enhanced learning ability in rats by showing its effect on cholinergic system and also due to its antioxidant effects. Singh and Dhawan 1982, Bhattacharya et al., 2000. In addition, bacopa extract showed potential anxiolytic, antidepressant and anticonvulsive action, Bhattacharya and Ghoshal 1998, Siaram et al., 2002, Shanmugasundaram et al., 1991. 31. Curcuma longa. Turmeric, Curcuma longa L. Zingiberaceae, rhizomes has been widely used for centuries in indigenous medicine for the treatment of a variety of inflammatory conditions and other diseases. Its medicinal properties have been attributed mainly to the curcuminoids, curcumin being its major component. Figure 5, Active curcuminoids major component of Hydra. Curcumin has been shown to possess wide range of pharmacological activities including anti-inflammatory, anti-cancer, antioxidant, wound healing and antimicrobial effects, Satosker, et al. 1986. Many of these biological effects of turmeric and its component are briefly illustrated in Figure 6, Radha, et al., 2006. Biological effects of turmeric/curcumin. Enhance. Wound healing. Immunomodulatory. Anti-inflammatory antioxidant activity. Turmeric/curcumin. Anti-mutagenic. Prevents IRI/neuroprotection. Antimetastatic. Antiviral, antifungal, antibacteria. Antiangiogenic. Figure 6, Multiple targets and functions of Curcuma longa and its active component curcumin. Hippophae rhamnoids, seabuckthorn. Hippophae rhamnoids L. Iliagnaceae, is naturally distributed over Asia and Europe. Different parts of H. rhamnoids L. have been used for the treatment of diseases in traditional medicine in various countries in the world. It contains a series of chemical compounds including carotenoids, tocopherols, sterols, lipids, ascorbic acid, tannins. However, flavonoids are supposed to be its most active bioconstituents, Figure 7. These compounds possess biological and therapeutic activity including antioxidant, antitumoral, hepatoprotective and immunomodulatory properties. Figure 7, Major bioconstituents of Hippophae rhamnoids, amyloidas, experimental design. 10C57BL/6 strain mice, 4 and 10 months old, were grouped in 3 groups. They had free access to pellet food and water in plastic cages and were kept 12H light, 12H dark cycle with room temperature 22 plus 1% C and relative humidity 50 to 70%. The experiments were complied with current ethical regulations on animal research of institution and all mice used in the experiments received humane care. Group I normal control, 4 months old mice. Group 2 diseased control, 10 months old mice. Group 3 aged mice treated with drug. Drug dose. 1. Brahmi, bacicides 20 mg per kilogram B. Weight. 2. Hydra, curcuminoids 15 mg per kilogram B. Weight 3. Amyloidas, Hippophae rhamnoids 18 mg per kilogram B. Wt. All the 3 drugs are orally administered for 3 months. Materials and methods. Bacicides, triterpenoid, saponins of Brahmi, Bacopa monnieri, have been extracted using N-butanol. The butanol layer is further extracted with ethyl acetate and both the soluble as well as insoluble fraction is collected. The insoluble fraction is extracted with chloroform, methanol, water, 20 hours 20 minutes and 60 seconds, which contains active bacicides. Drug is orally administered to animals for 3 months and at the end of the experiment animals are sacrificed by cervical dislocation and brain is quickly removed and kept at 85 degrees Celsius till further investigation. Similarly, curcuminoid's active polyphenolic fraction of Hydra, Curcuma longa, has been prepared by extraction with hexane and ethanol and finally precipitated with petroleum ether. Acetylcholine, noradrenaline and 5-HT estimation is performed by fluorometric method, Spieg et al., whereas dopamine estimation is done by method developed by Chang while acetylcholinesterase activity is measured spectrophometrically, Elman et al. The choline acetyltransferase, CHAT, activity is done by radiometric method of Haba K et al. And lipid peroxidase activity is analyzed by TBARS method, Okawa et al., total glutathione content is measured by method described by Elman et al. And catalase activity is measured according to AB using spectrophotometer, whereas glutathione peroxidase activity is detected by the method of Carrillo et al. Mitochondria is isolated in sucrose buffer containing 20 mm Heps, pH 7.5, 10 mm KCl, 1.5 mm MgCl, 1 mm EDTA, 1 mm EGTA, 1 mm dithiothiol, and 0.1 mm PMSF and 250 mm sucrose. Final pellet is collected from mitochondrial fraction and solubilized in buffer, which is used for immunoblotting experiments. Inflammatory markers are assayed in the SARA by commercially available ELISA kit, R&D System, USA. Experiment No. The aim of the present study was to investigate the protective effect of bacchicides on oxidative stress, mitochondrial dysfunction, inflammation and neurotransmitters concentration in aged mice. Results. Effect of Brahmi, bacchicides, on neurotransmitter and neurochemical markers in cortex of aged mice. Acetylcholine, NM/GM, dopamine, UG/GM, noradrenaline, μg/GM, 5-HT, μg/GM. Group I, asterisk, 26.52 plus or minus 3.58, 0.43 plus or minus 0.09, 0.43 plus or minus 0.14, 0.63 plus or minus 0.07. Group 2 foot asterisk, 16.72 plus or minus 2.61, 0.31 plus or minus 0.06, 0.18 plus or minus 0.06, 0.42 plus or minus 0.06. Group 3 asterisk, 23.57 plus or minus 2.91, 0.45 plus or minus 0.22, 0.35 plus or minus 0.22, 0.53 plus or minus 0.02. Asterisk, versus, P backslash 0.01. Asterisk, versus, P backslash 0.01. Effect of bacchicides on acetylcholinesterase activity, MM/GM tissue, in aged mice brain. Values are mean plus or minus SEM. Asterisk P less than 0.05 versus control effect of bacchicides on mitochondrial proteins in aged mice brain. NADH dehydrogenase, CYT C oxidase, B actin. A B C. A equals young, B equals aged, C treated.